The objectives of this talk are to understand the term maternal inheritance, to learn the cytoplasmic inheritance in plant Mirabilis jalapa, to know extranuclear inheritance in paramecium, to learn the mitochondrial DNA inheritance in peptide yeast and in human diseases, to understand the term maternal effect with example of snail shell coiling phenotype. Dear students, let me first introduce you to some of the basic concepts regarding this topic. In earlier programs, we have already studied about DNA, genes, chromosome and their role in inheritance. We have also understood about how genes flow from one generation to the next generation, which result into inheritance of specific trait of character. We have also learned Mendel's principles of segregation and independent assortment. These principles are based on the assumption that genes are located on the chromosomes, which are present in the nucleus of the cell. For most genetic characteristics, this assumption is valid and Mendel's principle allow us to predict the types of offspring that will be produced in a genetic cross. Although genes present on nuclear chromosome governs the inheritance, but these genes are not the only hereditary genetic material which is responsible for inheritance of the traits. Certain scientific evidence point towards presence of DNA molecules in the cytoplasm. In eukaryotic system, the extranuclear or cytoplasmic DNA molecule is present in specified organelles like chloroplast and mitochondria. This cytoplasmic or extranuclear DNA molecules along with their encrypted genes present in the form of mitochondrial DNA, chloroplast DNA or as a dispensable hereditary units have characteristic pattern of inheritance. They do not follow Mendelian inheritance pattern. The reason behind it is that the cytoplasm does not divide in a precise manner like chromosome during cell division, and moreover zygote inherits nuclear genes from both parents, but typically all its cytoplasmic genes come from only one of the gamete and it is usually the egg. A sperm or pollen generally contribute only a set of nuclear genes and not the cytoplasmic genes from the male parent. In this case, cytoplasmically inherited traits are passed from mother to offspring and never from father to offspring. The inheritance of these cytoplasmic genes encoded traits or inheritance of non-nuclear DNA molecules encoded traits are known as non-Mendelian, non-chromosomal, extrachromosomal, extranuclear, maternal, uniparental, cytoplasmic, mitochondrial or chloroplast inheritance. Cytoplasmically inherited characteristics frequently exhibit extensive phenotypic variation. As previously mentioned, the reason behind it is that no mechanism analogous to mitosis or meiosis ensures uniform distribution of cytoplasmic gene in cell division. Thus, different cells and individual offspring will contain various proportions of cytoplasmic genes. Let us take the mitochondrial genes as an example. Most cells contain thousands of mitochondria and each mitochondrion contain 2 to 10 copies of mitochondrial DNA. Suppose that half of the mitochondria in a cell contain a normal wild type copy of mitochondrial DNA and the other half contain a mutated copy. In cell division, the mitochondria segregate into progeny cells at random. Just by chance, one cell may receive mostly mutated mitochondrial DNA and the other cell may receive mostly wild type mitochondrial DNA. In this way, different progeny from the same mother and even cells within an individual offspring may vary in their phenotypes. Traits encoded by chloroplast DNA are similarly variable. There are number of well established examples of chloroplast and mitochondrial inheritance available, and some of the important ones are discussed here.
Let us see the inheritance of chloroplast gene and variegation in 4 o'clock plant, that is Mirabilis jalapa. In 1909, cytoplasmic inheritance was recognized by Dr. Carl Correns as an exception to Mendel's principles. Dr. Correns, one of the biologists who rediscovered Mendel's work, studied the inheritance of leaf variegation in the 4 o'clock plant Mirabilis jalapa. The first clue indicating something unusual in Mirabilis jalapa was that the inheritance of its leaf color phenotype did not obey Mendel's laws. In Mendel's experiments, it made no difference irrespective of which parent carried which phenotype. For example, a cross of pollen from a yellow seeded plant to the egg of a green seeded plant gave the same results as the reverse, that is, cross of pollen from a green seeded plant with egg of a yellow seeded plant showed similar results. In other words, we can say that if trait obeys Mendel's law, then reciprocal cross should show similar results. But with leaf color in Mirabilis jalapa, the results of reciprocal cross were different. That is, they did not follow the Mendel's law of inheritance. Dr. Correns found that the leaves and shoots of one variety of 4 o'clock plant were variegated, displaying a mixture of green and white color patches on leaf. He also observed that some branches of the variegated strain had all green leaves and other branches had all white leaves. Each branch produced flowers. So, he was able to cross flowers from green, white and variegated branches in all combinations. When Dr. Correns crossed any male, that is green, white or variegated type with a green female, the progeny were all green, and when he crossed any male with a white female, the progeny were all white. But when he crossed any male with a variegated female, the progeny were mixture of green, white and variegated with no fixed ratio. We can also explain it in another way, that the seeds from green branches always give rise to green progeny, no matter whether the pollen for that seed was from flower of a green, white or variegated branch. Similarly, seeds on white branches produced white progeny. Seeds on the variegated branches gave rise to green, white and variegated progeny in no particular ratio. In his experiment, Dr. Correns collected gametes from all white branches of variegated plants, since all white plants could not survive long enough to flower. Thus, it can be concluded that the phenotype of the male parent does not influence the leaf color of the progeny. It is the mother's cytoplasm which decide the color of leaf in the progeny. This pattern of inheritance is known as maternal inheritance. Now, let us see the example of extranuclear inheritance in paramecium by Kappa particles. In 1938, T. M. Sonneborn and his associates have described the transmission of some cytoplasmic particles known as Kappa particles and their relation to nuclear genes in paramecium aurelia. Individuals of a particular race of paramecium aurelia are called killer strain, which destroy other individuals of paramecium by secreting some water soluble toxic substance into the water in which they live. This substance is known as paramecin, and as mentioned earlier, such paramecium aurelia are called killers. On the other hand, the individuals destroyed by paramecin are known as sensitive. The paramecium of killer strain contain large number of Kappa particles in their cytoplasm. Like viruses, Kappa particles can duplicate themselves independent of the nuclear division. But in absence of chromosomal dominant gene capital K, Kappa particles cannot multiply. Moreover, in absence of Kappa particles, dominant gene capital K cannot produce them de novo. So, in the killer paramecium, the presence of Kappa particles, their maintenance and replication is dependent on the chromosomal dominant gene capital K. However, killer strain with genotype small kk cannot be obtained because even if Kappa particles are present, these would be lost in absence of chromosomal dominant gene capital K. When a paramecium of killer strain having the genotype capital KK conjugates with the paramecium of sensitive strain having the genotype small kk, the X conjugates are all heterozygous having capital K small k genes. The capital K small k genotype suggests that both X conjugates should be killers, but this is not the case. If conjugation is normal, that is, lasts only for a short time with exchange of only nuclear genes and no exchange of cytoplasm takes place between the two, both killers and sensitive are produced having similar genotype, that is capital K small k. However, rare or prolonged conjugation, that is lasting for long time, permits mixing of cytoplasm along with nuclear genes of both conjugates and produce killer paramecium only having capital K small k genotype. The killer type of phenotypic character is stable only in killer strain with capital KK or capital K small k genotype and is unstable in sensitive strain with small kk genotype. It is possible to convert killer clones having genotype capital KK into sensitive by making them undergo very rapid fissions. If paramecium of killer strain with genotype capital KK are overfed, they multiply very fast. On the other hand, the Kappa particles fail to multiply very fast and are unable to keep pace with the rapid fission of the paramecium body. Ultimately, it leads to loss of kappa particles entirely from paramecia which have genotype capital KK. The dominant gene capital K present in their genotype is unable to initiate the production of kappa particles, but can simply maintain them if these are already present or are introduced. Vice versa, the recessive gene small k is unable to maintain the kappa particles. If kappa particles are introduced into a sensitive paramecium having gene small kk, these are lost without producing any change in the phenotypic characters of the sensitive paramecium. Let us understand the maternal inheritance in petite yeast and in some human diseases by mitochondrial genes. Mitochondria from a variety of organisms ranging from yeast to humans are subject to alterations in their genetic material, that is DNA. These mutations cause phenotypic changes that are inherited in a non-Mendelian fashion. One classic example of a mitochondrial genetic defect first described by Boris Efroussi and his colleagues in 1949 affects the baker's yeast Saccharomyces cerevisiae. It causes cells to grow very slowly and to form small colonies. Therefore, afflicted yeast strains are called petite. The petite mutants are classified into following types: segregational petite forms which are developed due to mutation in the nuclear gene. They follow Mendelian segregation and hence are known as segregational petite. The non-segregational petite forms are developed due to loss or modification of mitochondrial genes. The cytoplasmic factor Rho is responsible for petite character and is located on mitochondrial DNA. There are two types of non-segregational petite. Number 1, neutral petite with no Rho factor, and number 2, suppressive petite with a defective Rho factor. Neutral and suppressive petite show non-Mendelian inheritance and are assumed to be caused by a cytoplasmic factor Rho. Is Rho really a mitochondrion? How do we know? Let us see that Efroussi showed the mitochondria of petites to be abnormal. They lacked some of the cytochrome proteins, that is A1, A3 and B, that help mitochondria to generate ATP. Since these cytochromes normally reside on the inner mitochondrial membrane, it is not surprising that petite mitochondria also showed underdeveloped inner membranes. It is also not surprising that yeast strains bearing such crippled mitochondria formed small colonies. If mitochondria cannot make ATP, the yeast cell must depend on the much less efficient process of fermentation or anaerobic respiration for its energy. This would naturally lead to slow growth. Moreover, due to dependency on fermentation for its energy, they are less sensitive or insensitive to inhibitors of aerobic pathway. Of course, these mitochondrial defects alone do not prove that the genetic lesion lies in the mitochondrion itself, because many mitochondrial proteins are encoded in the nucleus and imported into the mitochondria. Therefore, a defective mitochondrial protein more likely reflects a nuclear mutation than a mitochondrial one. However, after the discovery in the 1960s that mitochondria contain their own DNA, it became possible to compare directly the DNAs of normal and petite mitochondria. The differences are obvious, and it is suppressive petite mitochondrial DNA that has suffered rearrangements, deletions and duplication, and it is always incomplete. Neutral petite contain no mitochondrial DNA at all. These data strongly suggest that the mitochondrial DNA defects are a direct cause of the petite phenotype. Now, let us see some examples of human diseases that occurs due to mutation in mitochondrial DNA. In human, the mitochondria is inherited from mother through egg in the progeny. A number of rare human diseases that exhibit cytoplasmic inheritance due to defect in mitochondria have been identified. These disorders arise from mutations in mitochondrial DNA, most of which occur in genes encoding components of the electron transport chain involved in the generation of ATP in aerobic cellular respiration. One such disease is Leber hereditary optic neuropathy, abbreviated as LHON. Patients who have this disorder experience rapid loss of vision in both eyes, resulting from the death of cells in the optic nerve. This loss of vision typically occurs in early adulthood, usually between the age of 20 and 24, but it can occur any time after adolescence. Many missense mutations in the mitochondrial genes for the proteins like ND1, ND2, ND4, ND6, cytochrome B, etc. leads to LHON. These proteins are included in mitochondrial oxidative phosphorylation enzyme complexes. There is much clinical variability in the severity of the disease even within the same family. It appears that death of the optic nerve in LHON is a common result of oxidative phosphorylation defects, which is brought about by the inhibition of the electron transport chain. Leber hereditary optic neuropathy exhibits cytoplasmic inheritance, and the trait is passed from mother to all children, thus exhibiting maternal inheritance. Another example is myoclonic epilepsy and ragged red fiber disease. The affected individuals exhibit dementia, deafness and seizures. The mitochondria of these individuals are abnormal in appearance. The disease is caused by a single nucleotide substitution in the gene for tRNA for amino acid lysine. The mutated tRNA has a detrimental effect on mitochondrial protein synthesis, and it leads to various phenotypes of the disease. In this example also, the inheritance is from mother to child and irrespective to sex of the progeny. Additional example is of Kearns-Sayre syndrome, abbreviated as KSS, which is also known as oculocraniosomatic neuromuscular disorder with ragged red fibers. It is a mitochondrial myopathy with a typical onset before 20 years of age. People with this syndrome have involvement of the muscles controlling eyelid and eye movement along with encephalomyopathy. The cause of the syndrome is large deletions at various positions in the mitochondrial DNA. One model is that each deletion removes one or more tRNA genes whose products are essential for mitochondrial protein synthesis. This leads to development of the syndrome and essentially showing maternal inheritance. Now, let us understand about shell coiling of snail, Limnaea peregra, and maternal effects. From the examples studied so far, it can be concluded that maternal inheritance is controlled by the cytoplasm of egg, but there are situations in biology which show different type of inheritance known as maternal effect. Dr. Alfred Sturtevant in 1923 presented a different example of snail Limnaea. This water snail exhibits two different shell coiling phenotypes, dextral or right-handed by dominant allele capital D and sinistral or left-handed by recessive allele small d. When Dr. Alfred mated a homozygous dextral female having genotype capital DD with a homozygous sinistral male having genotype small dd, all the progeny were dextral like the maternal parent. Furthermore, when he mated a homozygous sinistral female having genotype small dd with a homozygous dextral male having genotype capital DD, all the progeny were sinistral again like the maternal parent. Is this not maternal inheritance? The answer is no, because it takes two more generations to discover exactly what is happening. In the next generation, Dr. Alfred allowed only the self-fertilization in the F1 progeny. This is possible because Limnaea is a hermaphrodite animal that contains both male and female reproductive organs and thus produces both male and female gametes. The F2 progeny did not resemble the maternal parent. Instead, they were all dextral. That does not sound like maternal inheritance. Neither does it sound like Mendelian inheritance. Finally, in the F3 generation, after all the F2s were allowed only for the self-fertilization, he could see a Mendelian pattern. There was a 3:1 dextral:sinistral distribution of phenotypes. Why did he not see this pattern in the F2 generation? Because the phenotype of snail depends on the genotype and not on the phenotype of the maternal parent, and dextral is dominant over sinistral. Cytological analysis of developing eggs has shown that the orientation of the mitotic division following fertilization controls the direction of coiling. It has been hypothesized that the mother encodes products that are deposited in the oocyte which direct the orientation of the mitosis spindle and therefore the direction of cell cleavage. Thus, a mother of genotype capital T small d or capital DD will deposit gene products that specify a dextral or right-handed coiling, and a mother of genotype small dd will deposit gene products that specify a sinistral or left-handed coiling. Support to this hypothesis has come from the experiments of G. Freeman and J. Lundelius. They injected cytoplasm from dextrally coiling snails into the eggs of small dd mothers who would normally specify sinistral coiling progeny and found that the resulting embryos coiled dextrally. In the reciprocal experiment, cytoplasm from sinistrally coiling snails was injected into eggs of capital D small d or capital DD mothers and had no effect on the resulting embryos. That is, they were still dextrally coiling. The interpretation is that the capital D allele specifies a product that is deposited in egg and causes the next generation embryo to coil dextrally, and the small d allele produces a defective or no product, so that small dd snails produce embryos that coil in the sinistral direction by default.
Dear students, now you have very well understood the differences between maternal inheritance and maternal effect on the basis of examples discussed in this talk. Beside this, here are some other examples of maternal inheritance and maternal effects which will further help you to understand this topic. Other examples of maternal inheritance are: number 1, extranuclear inheritance by mitochondria in Pocky strain of Neurospora; 2, male sterility in plant due to mitochondrial inheritance; 3, inheritance of IJEP characteristic, that is contrasting strips of green and white color on leaves by gene small I small j located on chromosome 7 in corn; 4, dispensable hereditary unit like sigma virus in Drosophila; 5, aminoglycoside antibiotic induced deafness, that is AAID, a human mitochondrial inheritance disorder. Other examples of maternal effect are: 1, in Drosophila embryogenesis, effect of expression of maternal bicoid gene; 2, in birds like Canaries and American Coot, mother hormones in egg affect an offspring's growth and behavior. Dear students, let us summarize the topic that we have been discussed so far. You know that inheritance has always been associated with nuclear genes. However, there are extranuclear DNA present in the eukaryotic cells within mitochondria and chloroplast, which do not segregate as nuclear DNA and thus do not follow Mendelian inheritance. The inheritance patterns of these cytoplasmic genes are known as non-Mendelian inheritance, non-chromosomal or extrachromosomal inheritance, cytoplasmic or extranuclear inheritance, maternal or uniparental inheritance, mitochondrial or chloroplast inheritance. Maternal inheritance is well understood with the help of classic example of leaf color in Mirabilis jalapa. Similar effect is also seen in Paramecium, where genotype of the organism and cytoplasmic particles are interdependent to express the killer phenotype. The mitochondrial gene mutation and its inheritance cause phenotypic or functional variation in the progeny of yeast, and it is also related to some human diseases. At last, the difference between maternal inheritance and maternal effect is understood by an example of snail shell coiling, which explains that maternal effect is different from maternal inheritance. Maternal effect is a phenomenon in which phenotype of a progeny depends only on the genotype and not on the phenotype of the maternal parent. Hope you fetched a good knowledge from this session. Thank you.